Hi, in this session you have chosen to study about the techniques of gene transfer into plants and animals. This module deals with the various techniques that are used to insert a gene of interest into host organisms, which are also called recipient organisms. Such a transfer creates transgenic organisms. When we say transgenic organisms, we mean only the plants and the animals and not genetically engineered microbes. The microorganisms into which new genes are inserted are called recombinant microbes and not transgenic organisms, as you must be already aware. Thus, this module will introduce you to the methods that are used to create transgenic plants and transgenic animals. After completing this module, you will be able to know the concepts of transferring genes from one organism to the other, comprehend the steps involved in the production of transgenic plants and animals, and learn the differences between the methods of gene transfer into plants and animals. As you are already aware, this module is part of the food biotechnology paper. It is designed to give you an overview of the experimental technologies used in the creation of transgenic organisms, including plants and animals. With this, you will be able to understand the differences between the physical, chemical and biological methods of gene transfer into plants and animals, resulting in the formation of transgenic organisms. Transgenesis, as you know from your last lesson, is the process of transferring and expressing a gene in a non-native cell or organism. It can also involve the removal of a native gene from an organism, resulting in what is called a knockout organism, or the exchange of one gene with another gene of known function, resulting in a knock-in organism. In animals, transgenesis also encompasses the transfer of an entire nucleus into an enucleated cell and developing it into an organism. This process is referred to as cloning. We shall see a little more information on these as we proceed further through the lesson. The newly inserted DNA sequence that leads to the formation of a transgenic organism is called a transgene. For successful integration and expression, a transgene should possess some specific components. They are a promoter sequence for the correct expression of the transferred gene of interest, the actual gene of interest to be inserted. This is the portion that is called the transgene. We also require a termination sequence to signal the end of the transgene, and finally, a selectable marker sequence to identify and select the successfully transformed cells or tissues. Together, all these components constitute the complete insert that is put into the host organism. Now that we are through with the basics, let us take up plant transgenesis. Let us start by defining a transgenic plant. A transgenic plant is one that contains either a single gene or a group of genes that have been artificially inserted into the plant instead of the plant acquiring them through natural process of pollination. The inserted transgene may come from various sources like another unrelated plant or from a totally unrelated species like bacteria or animals or even human beings. Now, let us move on to the methods of transferring genes into plants. There are several methods of transferring genes into plant cells. Broadly, they fall into three categories, namely physical methods, chemical methods and biological methods. The physical and chemical methods of gene transfer are also collectively referred to as vectorless gene transfer or direct gene transfer. The biological methods of gene transfer constitute the vector mediated gene transfer or indirect gene transfer. In these methods, the gene of interest is carried over to the target cell by a molecular vehicle called a vector. Thus, we have the physical, chemical and biological methods of gene transfer into plants. Let us consider them one after the other, starting with the physical methods. The major physical methods used in plant transgenesis include particle gun or gene gun, electroporation, and injection using a microscopic syringe, which is also called microinjection. Let us first take up the particle gun or the gene gun. This method of gene transfer is referred to as biolistic gene transfer. In this method, the foreign DNA containing the genes to be transferred is coated onto the surface of very minute gold or tungsten particles. These particles are only 1 to 3 micrometers in size. They are then bombarded onto the target tissue or cells using a particle gun. The particle gun is also called a gene gun or a shotgun or a microprojectile gun. We saw that the gold or tungsten particles are extremely small in size. For this reason, they are called microcarriers. In order to carry these microcarriers over to the plant tissue through the gene gun, bigger particles called macrocarriers, which are also called bullets, are used. The macrocarriers carrying the DNA coated microcarriers are loaded onto the gene gun and fired at the plant tissue, that is the recipient of the gene of interest. Though some cells are destroyed in this blast, many cells receive the DNA from the microcarriers. In fact, the biolistic method of gene transfer is very efficient in producing a large number of transformed cells and has been widely used to create transgenic plants. Two types of plant tissue are commonly used in particle bombardment, namely the primary explants, which are grown in culture, and proliferating tissue, which could be either embryonic tissues or the growing meristematic tissue. This method of gene transfer has been so successful that a pressurized handheld gene gun has been developed. This handheld device can be used to shoot DNA coated gold particles directly into the meristematic tissue of living plants that are growing in the experimental fields, converting them into transgenic plants. The second technique that we shall consider is electroporation. Electroporation, as the name implies, involves the generation of pores using an electric current. When pulses of high voltage electricity is applied to the plant cells, transient pores or holes are formed in the surface of the plant cell. If the plant cell is placed in a medium containing the DNA to be transferred, the DNA enters the cells through these pores into the cytoplasm and subsequently into the nucleus. Thus, we have successfully transferred the DNA from the medium into the cells. Generally, For the insertion of genes using electroporation, protoplasts are used. Protoplasts are nothing but plant cells which are devoid of the tough cell wall. Though the protoplasts are preferentially used, the technique has also been applied to whole plant cells as well as plant tissues. Now, we come to the next technique, namely microinjection. In this technique, the DNA is drawn into a very specialized nanosyringe or an even smaller femtosyringe. This is done with the aid of a microscope. In fact, the whole procedure is carried out viewing through a high definition microscope. The cell to be transformed is held in position using a nanoscale suction pipette, and the DNA is carefully injected into the cell. This way, we can ensure that the DNA is actually reaching the cell. In this technique, the DNA insert can also be prepared without the selectable marker gene, resulting in marker-free transgenic plants. Now, let us consider the methods of transferring genes using specific chemicals. Some chemicals can cause the instability of the cell membrane, resulting in the uptake of DNA molecules of interest. The commonly used chemicals include polyethylene glycol and dextran sulfate. Calcium phosphate has also been used in some studies, but the major chemical method that is used to employ liposomes. Liposomes or liposomes, as they are also called, are structures which are constructed with lipid molecules. They are spherical in nature with an aqueous core. If the liposomes are constructed in an aqueous medium containing the gene to be transferred, some of the genes are trapped in the interior of the liposomes. These DNA loaded liposomes can then be added to the plant cells, where they fuse with the membrane and release their contents or the gene of interest into the cell. For this technique also, protoplasts are preferentially used. As we have seen earlier, the biological methods of gene transfer are also termed as vector mediated gene transfer methods. The most common biological method used with plants is the agrobacterium mediated gene transfer. Agrobacterium tumefaciens is a soil bacterium that causes a tumor like growth called crown gall in the plants. This is the result of the transfer of a plasmid from the bacterium, and this plasmid is called the TI or tumor inducing plasmid. The DNA in this plasmid contains a portion called the tDNA. This tDNA is manipulated for the insertion of foreign DNA sequences. The tDNA is first cut open using restriction endonucleases, and then the gene of interest is inserted into it. The DNA is again closed and sealed using DNA ligase. When the plant cells are treated with the agrobacterium, which are containing the recombinant tDNA, by the natural infection process, the gene of interest reaches the target cell. For this reason, Agrobacterium tumefaciens is also called the natural genetic engineer. Another organism that is used for gene transfer into plants is Agrobacterium rhizogenes. Thus, using the physical, chemical and biological methods that we have discussed so far, several transgenic plants have been developed. Having learnt the methods of transgenesis in plants, we shall now see the same in animals. As in the case of the plants, there are once again physical, chemical and biological methods of gene transfer into the animal cells also. Coming to the physical methods that are used with animal cells, the major method of gene transfer into animals by physical means is electroporation. The principle of this method is the same as that employed for the plants. Since the animal cells do not contain a tough cell wall, the need for the preparation of protoplasm or other such structures does not arise, making it a preferred method that is employed with the animal cells. The use of gene gun to blast genes into animals, especially the mouse, has also been tried in some studies. The microinjection technique has been very successfully used to transfer transgenes into animal cells. However, more often the technique of microinjection in animal cells has been employed to transfer whole pronucleus rather than small pieces of DNA. In this method, a fertilized zygote is enucleated, that is, the nucleus is removed from the zygote by suction and in its place the nucleus from another cell is inserted by microinjection. This cell with the new nucleus is then inserted into the pseudo pregnant surrogate mother animal, which carries the calf to term and delivers it. The newborn calf will express the new genes in the transferred nucleus and is therefore the transgenic animal. Such a transgenic animal that is produced by the transfer of whole nucleus is more commonly called a cloned animal rather than a transgenic animal. Cloning is a technique that has been very successfully employed for the production of extinct species of animal and therefore is a much sought after method. Coming to the chemical methods of transferring genes into animal cells, polyethylene glycol or calcium phosphate can be used to destabilize the membrane for the uptake of DNA. The method of transfer is the same as that for the plant protoplasm. However, the more commonly used chemical methods of gene transfer into animal cells is the deployment of liposomes loaded with the gene of interest, as they readily fuse with the animal cell membrane and release the DNA into the cells. Several vector mediated gene transfer methods have also been devised for the transgenesis of animal cells. Of these, the most important ones are retroviral transfer, transgenesis by engineering embryonic stem cells, and transgenesis with artificial high capacity vectors. Starting with the retroviral transfer, retrovirus particles, which are highly infectious in nature, are modified to a defective state. This modification makes them lose the infective or pathogenic nature. They, however, retain their capacity to transfer DNA into animal cells. Helper virus strains, which can carry large pieces of inserted DNA, can also be used to transfer the genes. In this method, the animal embryo, usually at the 8-cell stage, is infected with the defective virus carrying the gene to be transferred. As the embryo grows, it expresses the gene of interest and is termed a transgenic animal. On the flip side of this method, sometimes the helper virus particle multiplies inside the animal cells. Therefore, the retrovirus mediated gene transfer is nowadays not the preferred method adopted for animal transgenesis. The next method is to use embryonic stem cells. During the development of the animal fetus, in the blastocyst stage, the cells can be harvested and maintained in culture. Such cells can proliferate very well under cultured conditions and still maintain their ability to differentiate into variety of cell types. For this reason, they are called pluripotent stem cells or simply embryonic stem cells or ES cells. Under cultured conditions, the embryonic stem cells are readily amenable to gene transfer. The acquired gene is expressed in these engineered cells. When these cells are reinserted into the blastocyst and introduced into the womb of a pseudo-pregnant foster mother, the embryo develops into a transgenic animal. Even though some of the methods discussed thus far are very effective in transferring short segments of DNA, they are inadequate to transfer large pieces of DNA, which is often desired in animal transgenesis. For this, high capacity vectors have been constructed by artificial means. These are collectively called artificial chromosomes. The major artificial chromosomes include BACs or bacterial artificial chromosomes, PACs or P1 bacteriophage derived artificial chromosomes, YACs or yeast artificial chromosomes, MACs or mammalian artificial chromosomes, and HACs or human artificial chromosomes. Of all these types, YACs or yeast artificial chromosomes are the most widely used vectors. The construction of YACs involves several steps. First, a circular bacterial plasmid DNA is cut open with a restriction endonuclease, resulting in a linear fragment. This linear DNA is then provided mitotic stability by inserting a centromeric DNA sequence. An origin of replication is then added to enable the plasmid to replicate extrachromosomally. Then follows the insertion of the artificial telomeric end sequences, which stabilizes the circular plasmid DNA into a very stable linear form. After this, the gene of interest is inserted using DNA ligase, making it a transgene. This recombinant artificial chromosome is then inserted into yeast for propagation and subsequent transgenesis into animal cells. Thus, in this module, you have learned about the various physical, chemical and biological methods of transferring genes into plant cells as well as animal cells. Such a transfer results in the production of transgenic plants and transgenic animals, as we have seen. The transgenic organisms have several applications in the field of nutrition as well as many other fields. We shall take them up in the next modules that we will consider. Though many applications exist and more are envisaged, the safety and the ethical aspects that are associated with the production and the use of transgenic organisms need to be borne in mind and addressed before more such organisms are devised. We shall consider them in the subsequent modules. Thank you.